For centuries, people noticed something peculiar about their hands. Sometimes the ring finger stretched longer than the index, sometimes not. But nobody knew why. Now, after decades of genetic research, scientists have finally cracked the code. Deep inside your DNA, on chromosome 6 and chromosome 14, two specific genes control this ancient difference, and what they reveal about you is far stranger than anyone expected. Put your hand flat on a table. Look at your second finger, the one you point with. Now look at your fourth finger, the one that wears a ring. Measure them. The difference between these two fingers is called the 2D/4D ratio, and it's one of the most studied measurements in modern biology. What started as a casual observation has become a window into prenatal development, athletic potential, cognitive ability, and disease risk. But for a long time, scientists couldn't explain why this ratio varied so dramatically from person to person. The story begins in the late 1800s when researchers first started documenting this finger-length difference systematically. They noticed a consistent pattern emerging across populations. Men, on average, had shorter index fingers relative to their ring fingers. Women's fingers were closer to equal in length. This observation seemed straightforward at first. Researchers assumed it was a simple inherited trait, maybe even controlled by sex chromosomes directly. But this assumption would turn out to be wrong in ways that nobody anticipated, leading scientists down a path that would eventually revolutionize how we understand human development. For decades, the mystery remained unsolved despite growing scientific interest. Scientists developed better measurement techniques, comparing X-rays of thousands of hands across different populations. They studied twins to understand how much of the variation was truly genetic and heritable. They looked at populations across the globe and found consistent patterns in the sexual dimorphism. Yet the fundamental question persisted unanswered. What actually controls this ratio? What determines whether your index finger will be longer or shorter than your ring finger? The answer, when it finally came, would require scanning the entire human genome and applying sophisticated computational analysis. In 2010, researchers conducted the first genome-wide association study, or GWAS, specifically designed to find genetic variants linked to finger-length ratio. This landmark study examined nearly 1,400 twins and their siblings from Australia. Using cutting-edge genetic technology and sophisticated association analysis, they scanned hundreds of thousands of DNA markers across the entire human genome. What they found was absolutely groundbreaking. A single location on chromosome 6, positioned inside a gene called Lin28B, showed a remarkably strong association with 2D/4D ratio. The specific variant, labeled as 314277, stood out with statistical significance of p equals 4, 1 times 10 to the minus 8. That's extraordinarily strong evidence in genetic research, the kind of finding that gets replicated and confirmed. Critically, researchers independently replicated this finding in an additional 5,129 children from the ALSPAC cohort. The association remained powerful even after controlling for height, which was important because height itself is influenced by many of the same genes. The confidence intervals were tight, the p-values remained far below the threshold for statistical significance. This wasn't a fluke. This was real. Lin28B was definitively linked to finger-length ratio, but this was just the beginning of the story. 3 years later, in 2013, researchers conducted another independent GWAS study in the United Kingdom, working with a completely separate population. This time, they identified a different location on chromosome 14 in a gene called SEMOC1. The variant here, labeled RS499-2-SED759, also showed powerful association with 2D/4D. Remarkably, the effect went in the opposite direction. The minor allele at SMOX1 was associated with decreased 2D/4D ratio, while the Lin28B variant was associated with increased ratio. This suggested that different genetic mechanisms were working in concert, pulling the ratio in opposite directions. When researchers combined the results from both studies through meta-analysis, the pattern became undeniable. Genetics definitely played a major role. But the complexity was immediately apparent. There were at least two distinct genetic loci influencing the trait, working through different mechanisms. The scientific community recognized that there was far more to discover. By 2018, scientists had pushed the research dramatically further. A comprehensive GWAS study published in the journal Human Molecular Genetics identified 9 additional genetic loci associated with 2D/4D ratio beyond the original 2. Researchers analyzed data from thousands of participants and discovered variants in genes including GLIS1, LDAH, OLA1, GLI3, FLI1, SAL1, TOX3, SAL3, and FNO1. The study also identified significant signals near genes involved in limb development like HOXD and HOXA clusters, though the strongest signals remained in the newly discovered loci. Suddenly, the picture became exponentially more complex than anyone had initially imagined. Finger length ratio wasn't controlled by one or two genes. It was influenced by at least 9 major genetic locations, each with subtle but measurable effects, plus potentially hundreds of smaller effects scattered throughout the genome. Most notably, the GLIS1 gene emerged as particularly important. This gene is involved in transcriptional regulation and cell growth. When researchers used a technique called depict to prioritize the most likely causal genes at each locus, GLIS1 stood out. The variants near GLI3 and FLI1 were equally compelling. These aren't random genes. They're all involved in developmental signaling pathways that regulate embryonic growth and patterning. This suggested that 2D to 4D isn't just about finger bone length. It's about fundamental developmental patterning mechanisms that get established before birth. Now, what makes this especially interesting is that genetics accounts for about 60 to 70% of the variation in finger length ratio between individuals. Twin studies have consistently estimated heritability between 50 and 80%, depending on the population and measurement methodology. That means the remaining 30 to 40% of variation comes from something else entirely. And that something else turned out to be one of the most important discoveries in the entire field. It's prenatal hormone exposure. During pregnancy, specifically in the second and third trimesters of fetal development, the developing fetus is bathed in sex hormones. Testosterone and estrogen flow through the maternal bloodstream and cross the placenta into fetal circulation. These hormones then permeate the developing tissues, including the bones of the fingers. The amount of exposure varies dramatically from fetus to fetus, influenced by maternal biology, placental function, and fetal genetics. Some fetuses experience very high testosterone exposure. Others experience very high estrogen exposure. Most fall somewhere in between on a continuous spectrum. This hormonal environment doesn't just pass without consequence. It actually shapes the physical structure of the developing hands, determining the relative lengths of the fingers that the person will carry for their entire life. The breakthrough was understanding how prenatal hormones interact with those genetic variants. LIN28B and SMOC1 aren't random genes selected by chance. They're both critical for early limb development and fetal growth. LIN28B is essential for embryo development and acts as a key player in stem cell development and proliferation. It's involved in regulating the timing of cell growth during critical developmental windows. SMOC1, meanwhile, is directly involved in regulating bone growth and the scaffolding of limb formation. These genes are active precisely during the critical window when prenatal hormones are most influential on developing tissues. That's where the connection lies. The genetic variants influence how these genes respond to testosterone and estrogen. And the hormones, in turn, regulate how strongly these genes express themselves. The most striking finding came from studying SMOC1 directly. Research showed that the protein encoded by this gene is upregulated by androgens, meaning testosterone activates it and makes it more active and abundant. The same protein is downregulated by estrogen, meaning estrogen suppresses it and reduces its activity. This is the intermediate mechanism. This is the biological link connecting prenatal hormones to finger length. A fetus exposed to high testosterone levels during the critical window would have more SMOC1 activity, which would influence bone growth patterns in the digits, resulting in a relatively longer ring finger. A fetus exposed to high estrogen would have less SMOC1 activity, resulting in a relatively longer index finger. LIN28B works through a similar mechanism. It's also responsive to sex hormone signaling during fetal development. The specific genetic variants alter how efficiently the gene can be activated or suppressed by hormonal signals. Someone inheriting a particular variant might have cells that respond very strongly to testosterone during development. Someone with a different variant might have cells that respond more moderately. These genetic differences get multiplied across millions of growing cells and millions of cell divisions during the critical developmental window, creating measurable differences in the final finger proportions. This mechanism explains why males and females show such consistent population-level differences. Due to chromosomal factors and endocrine system differences, male fetuses are typically exposed to significantly higher testosterone levels in the womb compared to female fetuses. That testosterone activates SMOC1 and other genes involved in differential growth. The result is that most men end up with ring fingers that are noticeably longer than their index fingers. Most women, experiencing lower testosterone and higher estrogen exposure on average, end up with more balanced ratios between the two fingers. But this is a population average. Individuals vary enormously. Some women have very low ratios, virtually identical to typical men. Some men have high ratios, similar to typical women. This individual variation is precisely what the GWAS studies were measuring. The implications of this discovery extend far beyond simple hand morphology. If the 2D/4D ratio reflects prenatal hormone exposure, and if prenatal hormones influence not just hand development, but also brain development, cardiovascular development, and immune system development, then the ratio could serve as a biological marker for many other traits and conditions. This hypothesis drove researchers to investigate correlations between 2D-4D ratio and everything from athletic performance to cognitive ability to disease susceptibility. The athletic research has been particularly compelling. A 2024 study examined the relationship between finger length ratio and oxygen consumption during incremental exercise testing. Researchers found that individuals with longer ring fingers, meaning lower 2D-4D ratios, metabolized oxygen more efficiently during maximal aerobic capacity tests. This suggested higher cardiorespiratory capacity. Another major study on female Olympic athletes compared their 2D-4D ratios to sedentary controls. The Olympic athletes had significantly lower ratios, suggesting that higher prenatal testosterone exposure was associated with athletic potential and competitive success. When researchers looked at specific performance metrics in the athletes, the 2D-4D ratio correlated negatively with strength tests and positively with endurance running performance. Recent 2025 research found significant moderate to sparse large positive associations between 2D-4D ratio and lactate production during intense incremental exercise. This means individuals with lower 2D-4D ratios, suggesting higher prenatal testosterone exposure, produced more lactate, indicating they could work at higher intensities before fatigue accumulated. This suggests that prenatal testosterone exposure influences muscle physiology at a fundamental level, perhaps affecting mitochondrial function or muscle fiber type distribution. But the relationship isn't universally simple or linear. A 2025 systematic review and meta-analysis examining 2D-4D and cardiorespiratory fitness found that the ratio is a reliable predictor of exercise tolerance and performance capacity, but any OT of aerobic efficiency or maximum oxygen uptake in all cases. The pattern that emerged suggests that prenatal testosterone exposure, as reflected in the 2D-4D ratio, influences motivation, pain tolerance, and the psychological capacity to push through discomfort during intense exercise. Individuals with lower ratios, those with higher prenatal testosterone exposure, may be more determined and better able to tolerate discomfort, which translates to better performance in high-intensity sustained activities. They might also have different training motivation or psychological profiles that lead to more consistent training and superior performance outcomes. Research on cognitive ability has produced more complicated and nuanced findings. Some studies reported that females with longer index fingers scored higher on cognitive tests at younger ages, consistent with better verbal abilities. However, older females with longer ring fingers performed better than those with longer index fingers on certain cognitive tasks. This nonlinear relationship appeared across multiple cognitive domains. Academic achievement, mathematical ability, spatial reasoning, all showed complex associations with 2D-4D ratio that varied significantly by age and sex. What this suggests is that prenatal hormones influence cognitive development, but not in simple ways. The effects might be different for different cognitive domains, and they might change across the lifespan as the brain continues to develop and mature. The behavioral correlations are perhaps most intriguing to researchers. Studies have linked 2D-4D ratio to personality traits and risk behaviors. Women with shorter index fingers, meaning lower ratios, suggesting higher prenatal testosterone exposure, were more likely to describe themselves as assertive and competitive. Men showed similar patterns. This makes biological sense. Higher prenatal testosterone exposure influences not just physical development, but also brain development in regions associated with aggression, dominance seeking, and competitive motivation. A December 2024 study identified an unexpected association between finger length ratio and alcohol consumption patterns, suggesting that individuals with longer ring fingers showed different drinking behaviors compared to those with longer index fingers. This finding hasn't been fully explained yet, but it hints that prenatal hormones might influence susceptibility to addictive behaviors or differences in reward processing. Health implications have attracted significant attention from researchers and clinicians. Studies have investigated potential links between 2D-4D ratio and various medical conditions. Researchers have examined correlations with obesity, migraine, eating disorders, depression, myopia, and autism spectrum conditions. The evidence is mixed, which is typical for complex traits influenced by multiple genetic and environmental factors. Some studies found statistically significant correlations. Others found weak or non-existent associations. What's become clear is that while prenatal hormone exposure is a fundamental biological factor affecting human development, it's not a strong determinant of most diseases by itself. The 2D-4D ratio is one variable among thousands that influence health outcomes. It's a marker, not a destiny. The cancer research deserves special attention. Some studies suggested that 2D-4D ratio correlates with risk for various cancers. Researchers hypothesized that because prenatal testosterone exposure influences immune development and cell proliferation patterns, it might affect cancer susceptibility throughout life. Prostate cancer and testicular cancer showed associations in some studies. Breast cancer and cervical cancer in women showed similar correlations. But again, the effect sizes were modest. The 2D/4D ratio alone could never be used as a predictive tool for cancer risk. It's one biological variable among thousands that influence cancer development over a lifetime. The 2D/4D ratio has also been studied as a potential biomarker for prenatal sex hormone exposure in investigations of neurodevelopmental conditions. Autism spectrum disorder has shown associations with 2D/4D ratio in some research, consistent with the idea that prenatal androgens influence brain development in fundamental ways. ADHD has similarly been studied. The prenatal androgen exposure hypothesis suggests that autism represents an extreme male brain phenotype, characterized by enhanced systematic thinking and reduced social intuition. This theory remains controversial and contested, but the 2D/4D ratio serves as one way researchers investigate whether prenatal hormones play a contributing role in the development of these conditions. What's remarkable about all this research is that the 2D/4D ratio is established before birth during fetal development and remains stable throughout the entire lifespan. It's not influenced by adult hormone levels. A woman in menopause has the same ratio as she did at 20. A man taking testosterone therapy doesn't change his ratio. The ratio is literally written into the bone structure during fetal development, fixed at that time, and remains unchanged. This makes it a unique window into the intrauterine environment, a biological fossil record of what that fetus experienced in terms of hormone exposure decades before. The measurement itself has its technical challenges, which researchers have worked hard to address over the years. Early studies asked people to simply look at their hands and determine which finger was longer by casual observation. This was imprecise because hand position and finger angle dramatically affect which fingertip extends further. The measurement varied depending on how flat you placed your hand, whether you extended your fingers fully, and the angle of view. Researchers developed more sophisticated techniques using calipers to measure from specific anatomical landmarks like the midpoint of the knuckle to the tip of the finger. Some studies used X-rays and high-resolution imaging to measure the actual bone lengths directly. Modern genetic testing companies like 23andMe and Ancestry DNA can now predict 2D/4D ratio using DNA analysis alone without any physical measurement at all. 23andMe researchers have identified 15 genetic markers that influence finger length. Ratio. They can analyze these DNA locations in a person's genome and provide statistical predictions about whether their index or ring finger is likely longer. It's not perfectly accurate because environmental factors and epigenetic effects continue to modulate the final measurement throughout development. But the predictions are statistically reliable and correlate well with actual measured ratios. Ancestry DNA offers similar trait analysis through their trait service. DNAgenics provides comprehensive 2D/4D ratio reports based on genetic data analysis. These companies are essentially performing personalized genomic prediction. The science reveals something profound about human development and the forces that shape us. We are not fixed at birth in obvious ways. Our intelligence, personality, and potential develop over time through countless interactions between our genes and our environment. But some things are fixed before we ever take our first breath. The ratio of our fingers is one of those things. It's determined by genetics and prenatal hormones, and from that point forward, it remains constant. It becomes a physical marker of that intrauterine environment, a biological reminder of the biochemical conditions we experienced before consciousness, before birth, before memory. This has philosophical implications that extend beyond pure biology and genetics. When we look at our hands and notice that one finger is longer than another, we're looking at evidence of a biological story that happened before we had any say in the matter. We inherited genetic variants from our parents that influence how our genes respond to hormones. We experienced a particular prenatal hormonal environment determined partly by our own genetics, partly by our mother's physiology, and partly by factors that science doesn't yet fully understand. That combination created a unique ratio of fingers that stayed with us for our entire lives. Our hands carry a record of our fetal development. The research community continues to uncover new associations and mechanisms. As GWAS studies grow larger and more refined with better statistical power, scientists continue to identify additional genetic variants. As epigenetics research advances, researchers are learning more about how environmental factors during pregnancy affect gene expression patterns. As neuroimaging studies become more sophisticated, scientists are mapping out precisely how prenatal hormone exposure influences brain development in different regions. The story of the 2D/4D ratio is far from finished. It's an ongoing frontier. One particularly interesting line of research examines what's called the 2D/4D asymmetry between hands. Some people have noticeably different ratios on their left hand compared to their right hand. Research suggests this asymmetry might reflect differences in prenatal hormone exposure between left and right sides of the developing body, or perhaps reflect different developmental timing between the two sides. Athletes often show asymmetric ratios, with some research suggesting that the side with the lower ratio, indicating higher prenatal testosterone exposure, sometimes shows better performance. This asymmetry is another frontier in the research that's beginning to be explored. There's also emerging work on what 2D to 4D ratio might tell us about other sexually dimorphic traits. Traits that differ between males and females systematically, like facial structure, voice pitch, bone density, and muscle mass distribution, might all be influenced by similar prenatal hormone. Exposure mechanisms. If 2D to 4D serves as a reliable marker of prenatal androgens, it could be used as a proxy variable in studies investigating how prenatal hormone exposure affects these other traits throughout development. The evolutionary perspective is equally fascinating. Why would evolution create this dimorphic pattern where males and females have such different finger ratios on average? Why would prenatal testosterone exposure influence finger growth at all? Most evolutionary biologists believe this is actually a byproduct of natural selection. The genes that respond to testosterone and produce the hormonal signaling system were selected primarily for their effects on other traits, like sexual development and reproductive success. The effects on fingers were largely incidental consequences. But over evolutionary time, different populations adapted to different environments, and the genetic architecture underlying 2D, 4D became more or less common depending on local environmental and social pressures. Cross-population studies have found consistent patterns in 2D, 4D ratios across human diversity. Males universally show lower ratios than females, though the magnitude of this difference varies slightly across populations. Some populations show slightly more dramatic sexual dimorphism than others. Researchers have found that populations living at higher latitudes sometimes show different average ratios compared to equatorial populations, though the reasons for these population differences remain unclear. It could relate to different patterns of prenatal hormone exposure across populations, or it could reflect genetic differences in the genes controlling 2D to 4D due to founder effects in population history. The measurement of finger length ratio has even become relevant in fields far removed from genetics. Anthropologists use 2D to 4D ratio to investigate ancient populations. Archaeologists measure finger bones from skeletal remains to estimate the prenatal hormone environment of people who lived thousands of years ago. This allows researchers to investigate whether there were historical shifts in prenatal androgen exposure across human populations over time, which might reflect changes in diet, environment, or social organization. One of the most interesting recent developments is the use of 2D, 4D in personalized medicine and sports science. As genomic testing becomes cheaper and more widespread, researchers are exploring whether 2D to 4D prediction from DNA could inform medical decision-making and treatment planning. Could someone's predicted 2D to 4D ratio, calculated from their genetic data, help clinicians predict risk for certain conditions? Could it inform sports science and athletic training recommendations? Could it help identify individuals who might be at higher risk for conditions like autism or ADHD, enabling earlier intervention? These are open questions, but the research infrastructure is actively being built. The complexity of the system is what makes it so scientifically compelling and intellectually interesting. A trait that appears simple at first glance, merely finger length ratio, actually involves at least 9 major genetic loci. Interactions with multiple sex hormones, epigenetic regulation during critical developmental windows, and environmental factors during pregnancy. It illustrates how even something seemingly straightforward in human biology actually reflects intricate molecular machinery operating during fetal development. Understanding 2D, 4D requires integrating knowledge from genetics, endocrinology, developmental biology, neuroscience, and environmental science, all working together in complex interactions. Perhaps most importantly, this research has humanized genetics in a way that few other discoveries have managed. Everyone can measure their own 2D, 4D ratio. Everyone can see the evidence of their prenatal history written into their hands right now. It makes the abstract science of genetics tangible and personal. It connects molecular biology at the level of DNA to something visible, something concrete, something you can literally see and measure. When someone looks at their fingers and realizes their ratio reflects prenatal testosterone exposure, which influenced their athletic capacity, their cognitive profile, and perhaps aspects of their personality, they're touching something genuinely profound. They're confronting the reality that who they are was shaped by biological forces that operated before memory, before consciousness, before choice. The research continues to evolve. Scientists are investigating finer details about the specific mechanisms. How exactly do SMARCA1 and Line 28B variants interact with hormone signaling? What are the precise cellular and molecular consequences of different genetic variants during critical windows of fetal development? How do the 9 confirmed genetic loci interact with each other? Do they have additive effects, or do they interact in more complex ways? These mechanistic questions will drive research for years to come. There's also growing interest in investigating the long-term health implications more thoroughly. Large prospective studies following people from birth throughout their lifespan with regular measurements of 2D-4D ratio, genetic genotyping, and extensive health tracking could clarify whether 2D.4D has genuine predictive value for disease risk, or whether associations found in smaller studies were statistical artifacts. Such studies are being initiated at major research centers. The story of finger-length genetics is ultimately a story about human development and the fundamental forces that shape us as individuals and as a species. It's a story about how our DNA interacts with our prenatal environment to create the physical forms we inhabit. It's a story about how childhood experience and fetal development create lasting imprints on our biology. It's a reminder that biology is precise, mechanistic, and operates according to rules we're slowly learning to read and understand. And it's a testament to human ingenuity that we develop tools sophisticated enough to decode these genetic signals, to understand mechanisms that operate in the darkness of the womb, to make the invisible visible through the patient accumulation of data and the relentless pursuit of understanding. Every time someone looks at their hands and sees their fingers, they're looking at a record of this extraordinary science, written into bone and tissue, a story that begins before birth and continues throughout life.